usual content, but I hope you guys enjoy. So, sit back and relax. Okay. Both local and long-distance signaling are used by cells to communicate. Examples of local signaling include direct cell-to-cell contact, as seen with the immune system, or by secreting local regulators. Growth factors and neurotransmitters are an example of this. Long-distance signaling involves chemicals, often released by one cell type that have traveled through the body to target cells, and another cell type. Hormones such as testosterone and estrogen function in long-distance signaling. There are 3 stages of cell signaling: reception, transduction, and response. Reception is the target cell's detection of a signal molecule coming from an outside cell. So, a good example of that is hormones. So the target cell first detects a signal molecule which comes from outside of the cell, so away from the cell wall, cytoplasm, whatever. Next is transduction. The binding of the signaling molecule changes the intracellular domain of the receptor proteins, allowing it to start transduction for the signals. The transduction stage converts the signal to form that can bring about a specific cellular response. So, the binding of the signaling molecule changes the intracellular domain of the receptor protein, allowing it to start transduction of the signal. So, in order for the signaling molecule to bind, it has to change the intracellular domain of the receptor protein, which starts the transduction of the signal. This stage converts the signal to form a response that can bring about a specific cellular response, so like insulin in the pancreas. What other example can I use? Okay, last thing is response. The specific cellular response of the signaling molecule. Is that all? Yeah. So it's like your blood sugar in response to eating food, like an hour later or whatever, 10 minutes later. The binding between a signaling molecule, ligand, and a receptor is highly specific. So, like, certain ligands cannot bind to other receptors. They are only made for one type. The conformational change in a receptor protein is often the first step in transduction of the signal. Receptors are found in 2 places. Intracellular receptors are found on the inside of the plasma membrane in the cytoplasm or the nucleus. The signal molecule must cross the plasma membrane and therefore must be hydrophobic. An example of this is testosterone enters the cell and binds to its specific receptor. This complex then enters the nuclei of a target cell. Plasma membrane receptors, or cell surface receptors, bind to water-soluble ligands. Example of this is the binding of epinephrine to the surface of a liver cell. Receptors cause a cascade that leads to the conversion of glucose in glycogen into glucose. A G protein coupled receptor is a membrane receptor that works with the help of a G protein. Step 1, the ligand or the signaling molecule binds to the G protein coupled receptor. The receptor is activated and changes shape when activated. The G coupled receptor binds a G protein that is in turn activated when it places GDP with a GTP molecule. Okay, so the ligand or signaling molecule binds to the G protein coupled receptor. The receptor is activated and changes shape when activated. The receptor binds a G protein that is in turn activated when placed in GDP with a GTP molecule. Step 2, the activated G protein leaves the receptor, diffuses along the membrane, and activates a specific enzyme where it binds to it. The activated enzyme triggers the next step in a pathway leading to cellular response. All the molecule shapes are changed temporarily, continuing the cell response to new signaling molecule. Okay, so the image, it shows the signaling molecule binding to the activated G protein coupled receptor, and that uses GDP and it activates the G protein, I'm pretty sure. And then it goes across the plasma membrane, the activated G protein, into the inactive enzyme where the GTP is basically powering the activated G protein to go into to be able to be binded to the enzyme. And when it is activated, it changes the enzyme shape, and that in turn creates the cellular response. Specific signaling molecules cause ligand-gated ion channels in a membrane to open or close, regulating the flow of specific ions. When the ligand opens to the channel and ions flow into the cell, the changes in the ion concentration can result in a change in cell activity. So, here we show a ligand-gated ion channel receptor in which the channel remains closed until a ligand binds to the receptor. So, only when the ligand binds will it open the channel and let ions into the cytoplasm, which is inside of the cell. When the ligand leaves, the channel closes again. Okay. Enduring understanding, blah blah blah. Cells communicate by generating, transmitting, and responding to chemical signals. Cell-to-cell communication is found in all biological systems and share across evolutionary lines of descent. Be able to describe how they receive signals and convert them into cellular response. Okay. So it says, the question says, describe how both G protein coupled receptors and ligand-gated ion channels receive signals and convert them into cellular response. So since the receptor, which is the G protein coupled receptor, and the gated ion channel, they have a specific binding spot for the signaling molecule to bind, and that in turn makes it cascade, I believe, so they can perform their function. When a ligand opens a channel and ions flow through the cell, the change in the ion concentration can result in a change in cell activity. So that's how it happens in the ion channel and in the G protein coupled receptor. The activated enzyme triggers the next step in a pathway leading to its cellular response. And to continue the cellular response, different molecules are needed. So that it's pretty well used. I might put a picture on the screen so you guys can understand what I'm talking about, because the pictures are much easier to comprehend than the words. I am a visual learner, so studying is not really my thing sometimes. Okay, signal transduction pathways often involve phosphorylation cascade. Phosphorylation cascade is the addition of a phosphate group into a molecule. Because the pathway is usually a multi-step one, the possibility of creating amplified the signal exists. At each step, enzymes called protein kinases phosphorylate and thereby activate many proteins at the next level. This cascade of phosphorylation is greatly enhanced. It enhances the signal and it allows for a larger cellular response. Protein phosphates are enzymes that remove phosphate groups and inactivate proteins that were activated by kinases. Thus, the signal can be turned on by kinases and turned off by phosphates. So, protein phosphates are enzymes which remove the phosphate group in the phosphorylation cascade. So the phosphorylation cascade is turned on by the protein kinase and then turned off by the protein phosphate, which removes the phosphate group. So that's pretty easy to understand. Not all components of signal transduction pathways are proteins. Many signaling pathways involve small non-protein water-soluble molecules or ion channels called secondary messengers. In the liver, the binding of the epinephrine activates cyclic AMP. Cyclic AMP and calcium ions are two common second messengers. The secondary messengers, once activated, can relay and amplify the signal, often by initiating a phosphorylation cascade, resulting in a cellular response. Many signaling pathways ultimately regulate protein synthesis, usually by turning specific genes on or off. That makes sense. This is a lot of this in this unit is very repetitive, just because it's just very simple. In some instances, changes in environment resulting in the signal transition pathway can alter the organism phenotype. In the cytoplasm, signaling pathways often regulate the activity of proteins rather than their synthesis. For example, the final step in the signaling pathway may affect the activity of the enzymes or cause the cytoskeleton rearrangement. An elaborate example of cell signaling in a program of controlled cell suicide is called apoptosis. During apoptosis, the cell is systematically dismantled and digested. This protects neighboring cells from damage which would occur at the dying cell. Blah 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 blah. Apoptosis is triggered by signals that activate a cascade of suicide proteins in the cell. In vertebrates, apoptosis is a normal part of development, and it is essential for a normal nervous system, for the operation of the immune system, and for normal morphogenesis of hands and feet. Okay, that's not important. Homeostasis is the maintenance and condition stability, blah 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 blah. I know what homeostasis is. Feedback mechanisms, I understand enough. If you don't know what a feedback mechanism is, it is a set point, and your body performs them in order to go back to that set point or to go further away from that set point. Positive feedback example is giving birth. So since the not having a baby is like homeostasis. It doesn't really make sense, but I can't explain that part very well. But negative feedback is like you have a fever, you sweat, and you go back to your normal temperature. Cell cycle is the life of a cell from the time it is formed. Blah blah blah. Mitosis is involved with growth and repair of tissues and asexual reproduction. A cell's endowment of DNA is its genetic information, also called its genome. Before the cell can divide, the cell's genome must be copied. All eukaryotic organisms have a characteristic number of chromosomes in their nuclei. Human somatic cells, all cells except gametes, have 46 chromosomes, in which the diploid chromosome number. Which is the diploid chromosome number. Diploid means two, by the way. Mitosis is the process by which somatic cells divide, forming daughter cells that contain the same chromosome number as the parent cell. Human gametes, sperm and egg cells, are haploid and have half the number of chromosomes as a diploid cell. Human gametes have 23 chromosomes. A special type of cell division called meiosis results in gametes. When the chromosomes are replicated, each duplicate chromosome consists of two sister chromatids attached by a centromere. One of the multiple chromosomes in a eukaryotic cell is represented here. Once it is duplicated, a chromosome consists of two sister chromatids connected along their lengths by a sister chromatid cohesion. Each chromatid contains a copy of the DNA molecule. Molecular and mechanical processes separate the sister chromatids into two daughter cells, which are genetically identical to the parent cell, unlike in meiosis. The two sister chromatids have identical DNA sequences. Later in the process of cell division, two sister chromatids will separate and move into new cells. Once the sister chromatids separate, they are considered individual chromosomes. Mitosis is the division of the cell's nucleus. It may be followed by cytokinesis, which is the division of the cell's cytoplasm. Where there was one cell, there's now two. Each the genetic equivalent. So when mitosis happens, it splits apart and they're exactly the same. The primary events of interphase, which is 90% of the cell cycle, follows. In the G1 phase, the cell grows while carrying out cell functions unique to its cell type. So, like, if you're an immune cell or if you're a red blood cell, that's a bad example. Like, if you're a liver cell, you'll be doing your regulation of whatever, detoxing the water, blah blah blah. In S phase, the cell continues to carry out its unique functions, but does one other important process. It duplicates chromosomes. This is called synthesis. I don't know why they don't have it written. This means it faithfully makes a copy of the DNA and makes up the cell's chromosome. In the G2 phase is the period after the chromosomes have been duplicated and just before mitosis. The cells continue to grow and carry out functions during this time. Mitosis can be broken down into 5 stages, not including cytokinesis, each stage. And prophase, the chromatids become more tightly coiled into a discrete chromosome. 2, the nucleoli disappear. 3, the meiotic spindle consisting of microtubules extending from the 2 centromeres begins to form in the cytoplasm. Prometaphase, the nuclear envelope begins to fragment, allowing the microtubules to attach to the chromosomes. The 2 chromatids of each chromosome are held together by the centromere. The centromere contains protein kinetochores on each chromatid, which is where the microtubules will attach. Metaphase, the microtubules move the chromosomes to the metaphase plate at the equator of the cell, which is the middle. The microtubule complex is referred to as a spindle. The kinetochores of the sister chromatids attach to the kinetochore microtubules coming out from opposite poles. This is very simple to remember. It's just PMAT. It's really PPMAT because you have prometaphase, but a lot of people don't learn about prometaphase. And the third part of metaphase is the centrales have migrated to opposite poles of the cell, riding along the developing spindle. So there's a lot of really cool pictures of like X-ray crystallography of like what this actually looks like, and it's honestly really cool. So I might put a picture if I can like find a nice one, maybe for my lecture or something. Anaphase is the next phase. Sister chromatids begin to separate, pulled apart by motor molecules interacting with the kinetochore tubules, and the cell elongates. By the end of anaphase, the opposite cells of the cell both contain complete and equal sets of chromosomes. Telophase, which is the last phase, the nuclear envelope. Really, cytokinesis is the last phase, but the nuclear envelope reform around the set of chromosomes. The chromatin fibers and the chromosomes become less condensed, so it just becomes another cell. Basically, that's what it looks like. Cytokinesis begins, during which the cytoplasm of the cell is divided in animal cells. A cleavage furrow forms. It's called that because it literally looks like a butt. It's really funny. That eventually divides the cytoplasm. In plant cells, a cell plate forms and divides the cytoplasm. The cell plate looks not as funny. Prokaryotes replicate their genome by binary fission. Bacteria do binary fission. Bacteria and other prokaryotes do not perform mitosis. Binary fission is actually like, I don't even get binary fission. Like, I learned it. I don't have it down in the notes because we didn't have to learn it right now. But it's like so confusing. If you didn't know, we were originally prokaryotes before we became eukaryotic organisms. Everything was basically evolved from prokaryotes. Prokaryotic division, which is binary fission. It's just like so complicated to understand. I don't know why. It's probably like way simpler, but like my poor head cannot understand it at all. The steps in the cell cycle are controlled by cell cycle control system. There are checkpoints during which molecular signals tell the cell to either continue dividing or to stop. The major cell cycle checkpoints include G1 phase checkpoint, G2 phase checkpoint, and M phase checkpoint, which is mitosis. The G1 phase checkpoint seems the most important if the cell gets to... If the cell gets the go-ahead signal at this checkpoint, it usually completes the whole cell cycle and divides. If it does not receive, then it goes into G0. Most mature human cells remain in G0 and never receive the molecular signal to divide. Muscle cells and nerve cells never divide, but liver cells can respond to signals moving from G0 back to the cell cycle at G1. Liver cells are a hot topic in this because they are dying a lot, I guess, maybe. I forgot there's like a real reason behind it. I think it is because they die more frequently. Don't drink alcohol, guys. Kinases are the enzymes that control cell cycle. They exist in the cell. So this goes back to the protein kinases used for the phosphorylation cascades. They exist in the cell at all times, but are active only when the connected regulatory cyclin proteins. Specific cyclin-dependent kinases give the go-ahead signals at the G1 and G2 checkpoints. They are also growth factors that will stimulate other cells to divide. A hallmark of numerous types of cancers and mutation in the kind is like in the kinase or the growth factor mutations. That's like what it's trying to say. Normal cell division has two additional key characteristics. Density dependent inhibition, the phenomenon in which crowded cells stop dividing. Anchorage dependent, C, in which normal cells must be attached to a substratum, such as the extracellular matrix of a tissue, to divide. So like binary fission, I don't think that has that, because if you don't know, all the like, well, I forgot the word. All of the little like organisms, not organisms, if you know what I mean, you know what I mean. They kind of just float around in prokaryotes. They aren't held down like in eukaryotic organisms. Knowing the features of normal cell division is important because cancer cells exhibit neither density-dependent inhibition nor anchorage dependency. Cancer cells exhibit a number of key mutations. A tumor is a mass of abnormal cells. I have done like I couldn't even tell you how many labs I've done on cancer within this past month. Like I can't take it anymore. Like it's just continuously like, and it's so repetitive. It's so fucking repetitive. Oh, I just cursed. It's so freaking repetitive. It's horrible. And I had a lab where you had to go over not every single obviously, but what they do for immunotherapy, where they look for a certain type of like cell protein thing, and you had to make a treatment plan for a patient based on like what type of cancer they had and what like protein thing they would be used for immunotherapy. I did not. My brain just was done. And then we had to do like some like spinach heart graft thing. It was horrible. Cancer cells are immortal, and there's no limit to the number of times they can divide, and they just keep on dividing within a space, as they do not exhibit anchorage dependent inhibition. If the abnormal cells are capable of surviving, and the new site is generally remained. If the abnormal cells are not capable of surviving at a new site and generally remain at the original site, the lump is called benign tumor. So benign tumor means not cancers. They like didn't explain that well. And a malignant tumor is cancer, basically, because they're able to invade and survive at new sites. The invading cells must reproduce aggressively. Metastatic cancer is when they invade to the blood or lymph vessels, and then they travel into different organs and different parts of the body. Okay, and that's all. Wow, that was like literally like 5 pages. And you know what? I literally knew all of it for some reason. But yeah, guys, please let me know if you guys like these types of videos, because I don't think I've ever done this before. So yeah, let me know if you guys like. Maybe one day I'll do a video of like me writing my notes out, because I do like to write them out on paper.